All right, good afternoon, everybody. I'm delighted to have you here, and I think I speak on behalf of the entire LA-HIP team that we're just really excited for you to come and share in the success of our students this summer. Every year I say that this is both my favorite and the most depressing day of the year for me at work, and it's really true, right? Because we've seen 24 students who've joined us for the summer for 7 weeks really blossom and advance their understanding of how to do research, learn to become a cohort with one another, learn a lot of things about themselves, and really just bring a lot of energy and joy to the institution. So we're delighted that they've made it this far and that they're going to get to share the amazing things they've done with you this summer, but then on Monday, they're not here. So, you know, happy, sad, I don't know, a little bit of both. But I'm really excited to introduce this class to you because they've been absolutely tremendous. Just the amount of enthusiasm, the level of the questions that they come and bring me in the office and that they ask their mentors in the lab about. It's really these are future leaders, absolutely no doubt. And so I'm just going to thank a few people and then get off of the podium because you're here to see them, not me. I absolutely want to make sure that we thank the fantastic LA-HIP staff. Danny, Emmanuel, and Mateo, could you guys stand up real quick? They really keep the program running, and also the entire TechPad team really pitches in to support at all kinds of times. Also the laboratory mentors, both the students, the postdocs, and the PIs have done a fantastic job this summer, and we couldn't do that without you, as well as all of our funding sources, and the folks who contribute to keep this thing going. You'll hear a little bit from him at the end today, but I want to once again thank Dr. B. Emil Bolgenman, who founded this program in 2005 and who's still comes back to do book club with the students, which is always a good time. And finally, I want to give a shout out to our long-suffering audiovisual technician, Ricardo, who really has made this so smooth this summer. He's been here for all of our classes, all of our practices, and now he's hiding in his booth. So, all right. So again, thanks for being here, and thank you that are online for joining us on the webinar. I'm going to introduce now our CEO, who's going to give us a few notes on his thoughts about the program and about how the education mission fits together here at CHLA. So, Paul Viviano, everyone.
Thank you, Dr. Fry. What a pleasure it is to be here this afternoon to have the opportunity to thank you and to congratulate you and to welcome you. So I have a few comments and then we'll try to move along quickly. So 19th Annual Science Symposium for the Samuels Family Latino and African American High School Internship Program, also known affectionately as LA-HIP. So I'm inspired by everything you have done and will continue to do, and we have some comments about the future, but it's a proud day for us to see the impact that you've had here at CHLA, and it's this history, this context that Dr. Bogeman founded in 2005. He had a vision, and that vision was to create more opportunities for those who are systematically underrepresented in medicine and science. And so nearly 2 decades later, LA-HIP is often a game changer for our students, our graduates, for these students as they enter their senior year in high school to help them develop their college applications and hopefully set their career on a great path. So in fact, over these past 19 years, every single LA-HIP graduate has gone on to enroll in a 4-year college. Can you imagine? Congratulations, what a magnificent record. Congratulations. And of course, many have remained in science and in medicine. We have PhDs and MDs among our graduates, and our alums come back proudly to celebrate with us. So this summer, you heard 24 students joined us here at CHLA, engaging in research areas, including regenerative medicine, gastroenterology, cancer and blood diseases, and neurosciences. So today, both in person and virtually, we get to hear the results of their work. And so I'd like to thank a few people. Again, thanks to Dr. Bogeman for his vision and his investment in this program all those years ago. Dr. Mark Fry, who has grown the program to its current size and level of expertise. You've heard about Danielle and Emmanuel. Thank you so much for your help and assistance. You've met Dr. Pat Levitt, who's the Chief Scientific Officer, timely for once. And just a little late. That's okay. It's perfect timing. Jody Ogden, who is our administrative director and leader for research. And so a couple of comments about how we got here. This program is funded by philanthropic support, the incredible generosity of the Samuels family, the Chuck Lorre Foundation, and the O'Connell Adams family, who have been so central to the commitment to this program. So I have a lot of friends in healthcare. And they tell me that I have the greatest job in healthcare. That may be debatable, but I can tell you I don't have the most important job in healthcare. The scientists, like Dr. Fry and Dr. Levitt, the physician scientists, the people that you've worked with this summer, they have the most important job in healthcare. They've devoted their careers, and we hope you emulate them. They've devoted their careers to scientific discovery, to developing new treatments and new cures for children, for providing world-class and compassionate clinical care to the precious children who are entrusted to our care, and to educate the providers, the scientists, and the caregivers of tomorrow, what this class represents. So thank you for being part of the Children's Hospital Los Angeles family. We hope that this summer will inspire you to pursue a career in science and medicine, to contribute, not just here locally, but contribute globally by developing those new treatments and new cures, to changing and transformative the healthcare that we provide to the precious children, not just in LA, not just in the greater LA area, but around the world. So thank you for being part of our family. You will always be part of the Children's Hospital Los Angeles family. You are always welcome here. We are amazed by what you've been able to accomplish. We thank you for your contributions. We look forward to your presentations this afternoon. Congratulations to every one of you and for their families. Thank you for supporting them. So I'll turn it back over to Dr. Fry. Thank you again. Thank
you. Okay, and with that, it gives me great pleasure to introduce one of our alumni who is going to talk about his journey to date. And well, we'll just do it. Oops. If I can get the computer to unfreeze, there we go. Okay. All right, Anthony Alvarado.
Good afternoon, everybody. Well, thank you for having me here today. I'm much an honor to be here. The last time that I was standing here behind this podium was actually 7 years ago. So today I will present my research paper or research topic on trusting the process, a journey from medulloblastoma, ADCC, antibody-dependent cell cytotoxicity research, to now the Charles Drew University, UCLA, David Geffen School of Medicine Medical Education Program, class of 2027. I'd like to thank Dr. Scarzade and his lab, and then also Cham and Long, who I'm not sure if they're with the lab anymore, but they were my mentors during the process. So who am I? I'm Antonio Alvarado. I grew up and still live in Wilmington, California, down in the South Bay of Los Angeles. I went to high school in the California Academy of Math and Science, also known as CAMS, class of 2018. And during the summer of LA HIP, summer of 2017, I was in the Scarzade lab doing some research on antibody-dependent cell cytotoxicity assays. And they had found some previous research, and they actually recently just published a paper on it 2 years ago. And so that was that. So my research question, what does the LA HIP vision look like? And for me, I think my hypothesis is that the LA HIP helps students acquire the early clinical experiences that they need in order to pursue medical or undergraduate education, excel in that, and then if they still continue and want to pursue, they can pursue MDs, PhDs, or MD PhDs. So methods, how did that look like for me? It looked like starting off in LA HIP, summer of 2017, and we went to Occidental College here with my cohort mates, Many of them are actually in medical school or PhDs now. And then here was me. I was practicing my research presentation like many of you guys did today, or like earlier throughout the summer. And then this was me 7 years ago, giving the same spot, same room, same spot. A little taller, a little different, but I'm there. And then these are my lovely mom and sister on right and left. And really, my sister's been like a second mom for me, so that's that. But what those pictures don't say is that that same summer, I was also battling brain cancer for the second time in my life. So the first time I was diagnosed when I was 9, and then just the summer, while I was actually applying for LA Hip, I was diagnosed with metastatic brain tumor. So I have 7 tumors in my brain and 1 in my spine, still to this day. And I remember talking with Dr. B, and he was like, hey, if you make it past surgery, are you willing to come? And I was like, I'll be there. As long as I'm functioning, I'll be there. So during that summer, I did some research on antibody cell cytotoxicity assays, and then I came back the following summer and volunteered in Dr. Scarzer's lab doing some tissue clearing work to see if we could image tissue without having to cut it, and that was a ton of fun. Then, thanks to the work of the Mosaic College Prep, especially Julie and Karen, I was able to go to the University of Rochester on a full-ride scholarship as a Handler Scholar, and so I graduated from there in 2022. Then, because of LA Hip, I had access to the NIH NIDDK Step Up program and did research actually in the South Bay across at Harbor UCLA in the LA Biomed Research Institute, now known as the Lundquist Institute. So I got to do some research there. And then I also, because of that, got to go to Maryland and give my presentation at the NIH, which was a ton of fun. And that summer, I worked in Dr. Suthan Cho's lab, and she's now at Caltech. Following that, I got into the NIH Buying Research Time program. Dr. B recognized that a lot of us kind of tried to pursue other jobs on campus in order to make ends meet, just for our own needs. And he created this program where we could do research and still get paid through the NIH. And then I also did LA Hip 2.0, the 2 summers after. I was actually supposed to do it in 2020, but we won't talk about that summer. So I did that program, and I also did the University of Rochester MSCP Summer Scholars Program. And following my graduation from the University of Rochester, I actually got a job in Pharma, Champions Oncology, doing research for a lot of different pharmaceutical companies. They would send us products that they thought had oncological applications, and we would just get random ID'd products, and we would test them in vitro and in vivo. The company had a whole pipeline for in vitro to vivo. And I was there for about 7 months in Maryland, Rockville, Maryland, just down the street from the NIH. Then I came back to LA and was working at Torrance Memorial, and I was working as a clinical research associate, working there really just enrolling patients and identifying patients that were good candidates for clinical trials, and that was really fulfilling. And I was only there I really wanted to be there for as long as I could until I got into medical school, but I was only there for 5 months. And that was because I actually got to go to medical school. I got accepted in May of last year, and this was me at my white coat. So this lady right here is actually my 4th grade English teacher, or my 4th grade teacher. She was my teacher when I was actually diagnosed with brain cancer. So she got to come to my white coat at UCLA. And then here, this is my white coat at Charles Drew, and the man right there standing to my right is actually my neurosurgeon who did my surgery back in 2010. And then this is my lovely cohort of 28 students. Without them, I wouldn't have made it this far in my first year. And then here is my family, really. My family here, they've been huge support to me, and now I look forward to have a little nephew. And this weekend, I find out whether I'll be having another nephew, or niece on the way. So that'll be exciting. And so overall, this is what the review of my methods look like. This is everything here is really all because of LA Hip. Without LA Hip, I don't think I would have been able to access any of this. Even just being able to wear my white coat today is a true honor. So for my results and conclusions, I got into medical school. The program works. And as I've mentioned before, truly my LA Hip experiences were foundational in order for me to access everything else that came after that. And as far as my future work for my project of life is to continue through the CDU UCLA program. And if the fate works in my favor and the match, which is a computer deciding my future, I will hopefully match into neurosurgery and then maybe end up back here at CHLA doing a fellowship of some sort. But it doesn't end there. I also specifically wanted to shout out, this is actually Melanie. Melanie Morales was in the LA-HIP class of 2018, the summer after. And this past year I worked with her. She reached out to me and I mentored her through the application process for medical school. And she just actually began medical school in the new Charles Drew medical degree program. So, yeah. So to all the students, you all have my information. When the time gets there, reach out. I'm always happy to help. If I don't respond to your email, email me again. I've gotten bad. I guess the neurosurgery tendencies are building on me already. Thank you.
All right, thanks a lot, Anthony. Terrific. Are you going to publish this? All right, and so now I'd like Danny Nunez to come up and introduce you to the students and shepherd us through the next hour of exciting science.
Okay, thank you for that intro. I am so happy to be here with you all. This summer has been so exciting, so rewarding, and just watching all 24 of them present this morning made me a little emotional, but we're going to do it again right now. I will be kind of introducing the students as they come up and present their research, and then, yeah, we can kind of roll in. All my students, big breath. Thank you. Okay, so I would love to welcome up Obi and Bella. Obi attends Fairfax High School, and Bella attends Girls Academy Leadership Academic Leadership Academy Gala. My bad. For Obi, she loved her favorite part of LA-HIP was that she loved learning about the kidney and was able to gain really valuable knowledge in the lab. Bella also really enjoyed the community that was fostered in the LA-HIP lab and is looking forward to implementing her passion for research in her career. So I'd love to welcome up Obi and Bella. It's okay. It's okay. You're so much taller.
Hello, my name is Isabella Kraus.
My name is Obio Yoga.
And today we'll be presenting our research on the use of stem cell-derived extracellular vesicles to modulate mitochondrial function in glomerular endothelial cells.
The kidney works to filter your body by removing waste from blood through urine and balancing the body's chemicals. When the kidney is dysfunctional, the waste in the body builds up and affects the nephron, which is the functional unit of the kidney. This leads to proteinuria, an elevated level of protein in the urine, a symptom of Alport syndrome or AS. AS is a hereditary form of chronic kidney disease or CKD and does not have a current cure.
The glomerular filtration barrier, or the GFB, consists of three parts: the glomerular endothelial cells, also known as JEC, the glomerular basement membrane, also known as the GBM, and podocytes. A dysfunctional GBM leads to JEC damage. JEC damage occurs in early stages of Alport syndrome due to a dysfunctional GBM and elevated vascular endothelial growth factors, or VEGF.
So, the elevated levels of VEGF induce mitochondrial damage in JEC. This image reflects in an animal the Alport syndrome continued to progress. So the mito tracker, which is the green, stains active mitochondria in the cell. PD tomato stains the endothelial cell. So as Alport syndrome continues to progress, the mitochondrial count shown in the green decreases and the endothelial cell count shown in the red increases. We seek to reverse this damage with the use of AFSC. Amniotic fluid is a liquid that surrounds the fetus during pregnancy. Amniotic fluid stem cells, or AFSC, is used to exude extracellular vesicles, or EVs. EVs are nano-sized particles from the cells that consist of molecules that's similar to its origin.
This leads to our research question. Can EVs from amniotic fluid stem cells be used to treat Alport syndrome? We hypothesize that amniotic fluid stem cell-derived EVs can protect against VEGF-induced mitochondrial damage.
To start our experiment, we cultured amniotic fluid stem cells in serum-free conditions for 24 hours before harvest. This culture was then turned into a supernatant through centrifugation and ultracentrifugation to remove content that is not extracellular vesicles, or EVs. With this, the EVs are then taken to nanosite tracking analysis to calculate the number of EVs in our sample and confirm its purification.
We then created 3 groups in concentrations of both 300 and 400 nanograms per microliter with 10,000 EVs in each well. These groups were examined at timeframes of 24, 48, and 72 hours with 1 triple. We then had a treatment group. Our control group contained JEC cells along with our 2 treatment groups. Our first treatment group contained JEC and VEGF to examine VEGF-induced JEC damage, and our second treatment group contained JEC, VEGF, and EVs to examine how EVs can reverse VEGF-induced JEC damage. We extracted the proteins from these samples and ran Western blot to quantify these differences. The resulting Western blot showed significant differences for the mitochondrial marker ATP5A, which is responsible for ATP synthase. VEGF stimulation at low dosage did not affect ATP5A levels in endothelial cells. Contrary to our hypothesis, EVs had the ability to downregulate ATP5A. In contrast, VEGF at high dosage significantly decreased ATP5A levels. This concludes that the effect of VEGF on mitochondrial enzymes is dose-dependent. Even at a higher concentration of VEGF, EVs were not able to reverse VEGF damage for the marker ATP5A. We also tested TOM20, a different mitochondrial marker, and in both low dose and high dose of VEGF, there was no significant effect. However, interestingly, upon multiple stimulations with low dose of VEGF, TOM20 expression was highly elevated, yet returned to baseline levels as a result of EV treatment. These results suggest that VEGF has a variable effect on mitochondrial genes. Due to the variability in VEGF's concentrational effect on mitochondrial genes, we wanted to investigate if VEGF was also time-dependent. To study this, we repeated the same experiment, yet instead of Western blot, we stained our cells with MitoTracker and analyzed these results with flow cytometry.
These 2 graphs show the same time frame. So originally, our experiment was to understand in depth how VEGF, or vascular endothelial growth factors, affect mitochondria. Since the effect of mitochondria is time-dependent, this is so we can refine our future experiments, take into consideration the specificity and details of JEC and the VEGF relationship. From this, we have learned that the effect of mitochondria is indeed time-dependent. We conclude that VEGF has a short effect on mitochondrial expression. In conclusion, VEGF affects mitochondrial function as confirmed by experiment. EVs can modulate VEGF-induced damage to the mitochondria. Future research must be done toward improving EV therapy to prevent endothelial damage, thus preventing the progression of CKD and Alport syndrome. Thank you to our friends on the 5th floor and the amazing community and this opportunity.
Thank you.
Thank you, you two. Now I would love to welcome up Pedro and Anthony. Pedro went to Alliance Schmidt Tech, and Anthony is from Harbor Teacher Prep. They both really enjoyed the program this summer, and Pedro notes that I like that LA HIP builds community among its students and alumni. What I look forward to is my future. Sorry, this is a good one. To do in my future is to ride a Type 2 VW bus on my way to my TED Talk. And Anthony says, I love the relationships and experiences I have gained, and I hope that this boosts me as a candidate when applying to colleges in the fall. So I'd love to welcome up Anthony and Pedro.
Hi, everybody. My name is Anthony Montijo,
I'm Pedro Olmedo.
and today we're going to be exploring the significance of fibrous collagen in fusion-positive rhabdomyosarcoma.
To begin, what is rhabdomyosarcoma? Basically, rhabdomyosarcoma, or RMS, is a very malignant pediatric sarcoma, really a type of cancer affecting soft tissue, muscle tissue, bones, and joints. The image you see on your left is a type of rhabdomyosarcoma called fusion-negative rhabdomyosarcoma. Basically, this type of sarcoma is less aggressive, more common, as no fusion gene.
Fusion-positive rhabdomyosarcoma. This subtype tends to be more aggressive and less prominent among the two subtypes. The factor that highlights the key difference between these two subtypes is the PAX3 FOXO1 fusion gene. This fusion is a result of a reciprocal translocation of chromosomes 2 and 13, which joins the DNA binding domain of PAX3 with the transactivation domain of FOXO1. This produces the PAX3 FOXO1 fusion protein, which is a transcription factor that drives cell proliferation. Now, as you can see in this image, it looks a little different from the fusion negative. You see these collective cell masses, and as you can see, these marginal or outer cells are interacting with this collagen, while these submarginal or inner cells aren't getting that same interaction. This leads us to ask, how does the interaction with fibrous collagen correlate with the PAX3 FOXO1 fusion gene? We hypothesize that there will be a lower expression of PAX3 FOXO1 in the marginal cells than the submarginal. In order to investigate this, we used a Western blot in which we seeded a fusion-positive rhabdomyosarcoma cell line onto different concentrations of collagen, and we isolated the protein of interest, which in this case is PAX3 FOXO1, and we were able to move our proteins onto a membrane using electrophoretic transferring. We then developed the membranes, and we're pleased with our results. So, like I said, we used a fusion-positive rhabdomyosarcoma cell line, RH30, and here we have our control GAPDH, where you can see even expression levels throughout. However, when we come to our PAX3 FOXO1, we see as the concentrations of collagen increase from 0 to 10, we see that the expression levels of PAX3 FOXO1 start to fade away. This supports our hypothesis that PAX3 FOXO1 expression levels are lowered in the marginal cells interacting with the collagen.
So moving on, this leads us to our next question. How do the different expressions of PAX3 FOXO1 affect the role of marginal and submarginal cells? This leads us to our hypothesis of the marginal cells are responsible for invasiveness, while submarginal cells are responsible for proliferation and replacing those cells. To test this, we did a hang and drop method where we added little cells onto a tissue culture to this, hang them, to flip them upside down and put them into incubator. From there, we observed highly grown collagen type 1 under a confocal microscope. So this right here is a video of that interaction. As you can see, the outside cells, the marginal cells, interacting with the collagen and started to invade the collagen. To add on from this, we decided to stain our spheroid with DAPI and SMAD3. DAPI is a nuclei is a marker for nuclei in DNA, and SMAD3 is a marker for cell invasion. As you can see in red, SMAD3 is on the outside or in the marginal spheroid, meaning that it will interact with the fibrous collagen. As supported by antibody results, this will decrease PAX3 FOXO1 expression and cause these cells to invade. In addition, we decided to again stain our spheroids, but instead for SMAD3, we are looking for PAX3 FOXO1. PAX3 FOXO1 in red marks cell proliferation and is inside our spheroid. This means that it will not interact with the fibrous collagen, and instead, as supported by antibody results, the expression of PAX3 FOXO1 will not decrease. This will lead the cells to proliferate and grow.
So the results of our experiments support both of our hypotheses that the interaction of fibrous collagen decreased the PAX3 FOXO1 expression, and marginal cells are responsible for invasiveness, while the submarginal cells are responsible for proliferation. In order to investigate this further, we can explore different signaling pathways that may be disrupted along the expression with PAX3 FOXO1 regarding the different behavioral patterns of the cells. And we want to give a special thanks to Dr. Park's lab. Thank you.
Thank you, Pedro and Anthony. Now, I am so excited to introduce Gianna and Nuif. Gianna goes to Linda Marquez High School, and Nuif goes to Culver City High School. Both really appreciated the hands-on lab experience that they had and the collaborative work that they did with their mentors this summer. So, turn in the microphone.
Good afternoon. My name is Nuif Dañacho.
And my name is Gianna Anguiano.
And our presentation is on the role of FAM174B in zebrafish cardiovascular development. So zebrafish are a known model for regeneration, just like Deadpool. As we know, the heart is a vital organ responsible for pumping blood throughout the body, supplying oxygen and nutrients while removing waste products. Its, its structure and function are crucial for the overall health. Zebrafish are a commonly used model because of the ability to study them during embryonic development. Notably, they are known to possess the ability to regenerate tissues and organs after injury, one of them being the heart, which can be regenerated within 90 days. Despite their simpler structure, zebrafish are ideal models for studying cardiac development and function. This is particularly relevant in the Lian lab, where regenerative medicine is used to study the heart following injury.
So in the heart, there are two important components. The blood vessels are responsible for carrying waste and carbon dioxide away from the tissues and organs, as well as carrying blood to them. The lymphatic system is responsible for making sure that the body has a balanced amount of tissue fluid. In a previous study done by the Lian lab, it was seen that after cryo injury, there was an increased amount of vessel growth. The reason that the Lian lab became interested in FAM174B was through a single-cell RNA of the bulbus where FAM174B became a candidate. With that being said, there is not much known about the FAM174B gene, but in humans, it is a response to stress protein that is required for the Golgi apparatus integrity. Through further investigation and the use of the human atlas, it was seen that in both humans and zebrafish, the FAM174B gene was located in the cytoplasm. There is a conserved function in the human and zebrafish, but it is unknown as to where it is exactly expressed.
So, building on this foundation, our research question is, where is FAM174B expressed and what is its role in cardiovascular development? We hypothesize that FAM174B is expressed in the blood and/or lymphatic endothelium, and loss of FAM174B function will compromise vessel development. So, in order to test our hypothesis, we utilize pre-existing data from ZebraHub to identify appropriate gene markers to use for our study. We validated the expression of FAM174B in the human and in the zebrafish. To distinguish between wild-type and FAM174B mutant fish, we performed genotyping and PCR analyses. To visualize the hearts, we used microscopy and embedded the samples in paraffin. Using a microtome, we sliced and embedded the heart tissue and mounted the sections onto slides. This enabled us to conduct further examination using fluorescent microscope, allowing us to analyze the expression of FAM174B in the context of cardiovascular development.
So as mentioned in our approach, we began our experiment by using the ZebraHub atlas to compare various marker genes to the FAM174B gene. Our analysis revealed that FAM174B exhibits similar expression patterns to other genes, specifically ETV2 and FLIP4, as shown in the images showing their similar locations. Notably, ETV2 and FLIP4 are recognized for their roles in the lymphatic system and endothelium, which suggests a potential functional relationship which should be further investigated. These images represent immunofluorescence using cells of the human FAM174B gene, which helps us confirm the conserved function between human and fish. In the images shown, it shows FAM174B expressed in the human lymphatic endothelium. To further examine these cells, the blue stain was DAPI, which represents the nucleus. The green stain is a representative of FAM174B, and the red is ARF1, which represents the Golgi.
This graph illustrates the percentage of area lacking ETV2, indicating regions without endothelium in the ventricles. As shown, the control group exhibits less than 10% of the ventricular area absent of ETV2, which is significantly smaller than the FAM174B mutant group, where 50% of the ventricular tissue is missing. This is visually represented on the images on the right, where the green staining indicates the ventricle, revealing that FAM174B mutant exhibits less density. This distinction is crucial because it highlights the role of FAM174B in endothelial development and suggests that mutations in this gene can lead to substantial defects in ventricular structure, potentially impacting heart function and overall cardiovascular health. So to summarize our results, firstly, through using ZebraHub, we were able to discover that FAM174B shows expression in the endothelium and lymphatics. Additionally, through our immunostaining, we observed that FAM174B mutant has decreased ventricular vascular coverage. Moving forward, our primary focus will be on deciphering the role of FAM174B in the endothelium. Moreover, we will evaluate the potential role of FAM174B in cardiac regeneration.
We would like to thank the Lian Labs, CHLA faculty, LA HIP, and our mentor Lau for guiding us through these 7 weeks. Thank you.
Thank you. And now I would love to welcome up Melanie and Alexa. Melanie attends Girls Academic Leadership Academy, and Alexa attends Teach Tech Charter High School. Melanie said she loved most about LA Hip was that through her internship, her favorite thing was working with BON 4 cells. I'm going to let them talk about that one because that's a little out of my wheelhouse. But she says, I love how confident I am in cell passaging or working in the hood. I am beyond excited to start my career in college by joining these research internships and exploring her passion for science at a college level. Alexa says that she loved having the opportunity to work in the lab with amazing people who are committed to teaching overall lab information and investing in both her and her partner's success. So I would love to invite those two to the stage.
Good afternoon. I'm Melanie Simone.
And I'm Alexa Torres.
And our research project is focused on inducing neurogenin 3 in BON4 cells to model pancreatic progenitor maturation. The prominent disease, type 1 diabetes, derives from beta cell dysfunction. Beta cells in the pancreas regulate endocrine functions like blood sugar. Type 1 diabetic patients face a lifelong condition where they are unable to create insulin due to their lack of beta cells. The current treatment for type 1 diabetes is not a long-term solution and is costly. Islet donors for beta cell transplants are limited and are not a long-term solution. Moreover, insulin therapies can be a financial burden and can decrease the quality of life.
However, a promising treatment for type 1 diabetes patients depends on improving beta cell therapies, which to improve we must go through the pancreatic stage first. Our research focuses on the beta cell differentiation, specifically the pancreatic progenitor phase. The field has established that the transcription factor neurogenin 3 is required for the transition from pancreatic progenitor to endocrine precursor. However, preliminary data in Georgia labs suggests that it is not only necessary for the endocrine progenitor stage, but the development of pancreatic progenitors. We found this via a CHLA patient who had a null mutation in their neurogenin 3 and they could not create pancreatic progenitors, suggested that it was needed for that specific development. Our transgenic cell model can help us better understand the pancreatic progenitor maturation to improve stem cell therapies.
We are using BON4 cells, a pancreatic carcinoma cell line, to model pancreatic progenitors. We are also working with cells that have been integrated with an inducible neurogenin 3 plasmid, and by adding doxycycline, we are expecting to see neurogenin 3 expression. This will work to test the gene and validate that the system we gave it functions.
So we asked, can we measure doxycycline-inducible neurogenin 3 activity from a transgenic BON4 cell line? So we hypothesized that the presence of doxycycline will activate human neurogenin 3 expression, its target NeuroD1 in the qPCR, and decrease cell proliferation in the EDUSA within the transgenic BON4 cell line.
We measured the gene expression of NeuroD1, a neurogenin gene target, through a qPCR. We started with maintaining our BON4 cells and using doxycycline to induce neurogenin 3 expression. We then extracted the RNA, moved on to cDNA synthesis, and then analyzed the qPCR results.
Then we branch off into measuring the proliferation rate of our cells with an EDUSA. First, we incubated our cells for 4 hours with an EDU solution, and then we fixed our cells, which we then labeled them with EDU, so once we get to imaging, we can see the proliferated cells as green compared to the total cells that are stained blue with host.
When compared to our BON4 control and our non-induced cell line, an increase in NeuroD1 is seen. This suggests that neurogenin 3 is being expressed and is binding to its target gene, NeuroD1.
So we use host for our total amount of cells, mCherry for to identify our plasmid, EDU for our total proliferated cells. So with our regular BON4, we see that we don't have an integrated plasmid, so we also didn't see any change within its proliferation because there's nothing to activate. But with the BON4 with the integrated plasmid, we see that there's quite a huge decrease with proliferation. It went from around 35% down to 8%, and this means that our cells are differentiating, meaning that our system is working.
In all, our hypothesis is supported. To start, neurogenin 3 is expressed within our transgenic cell model with the inclusion of doxycycline.
And there's a twofold increase of NeuroD1, a neurogenin 3 gene target, as seen through the qPCR results. And the EDU assay highlighted the decrease in cell proliferation in the transgenic cell line induced with doxycycline.
Lastly, studying the function of neurogenin 3 in pancreatic progenitor development can help in developing more efficient and long-term beta cell therapies for type 1 diabetes patients. To the Georgia Lab and the following institutions and programs.
And we'd like to thank Dr. Georgia for the opportunity to work in her lab and to our amazing mentor, Marnie. Thank you. Thank you. Now I would like to welcome up Sophia and Michelle. Sophia attends John F. Kennedy High School, and Michelle attends Applied Technology Center. One of Sophia's favorite parts of LA HIP, as well as Michelle's, was the hands-on lab work that they were able to do and the connections that they were able to build in the lab as well. Michelle says that I loved getting to meet people from very different backgrounds every day and exploring my interests. This has made me look forward to being able to continue research in neuroscience, as well as being the first person in my family to attend a four-year university because of the mentoring through LA HIP. It is now my pleasure to welcome up Michelle and Sophia.
Hi, my name is Sophia Moraz.
And I'm Michelle de la Cruz.
And today we're examining CHLA expression within the metaprefrontal cortex across two genetic backgrounds. So, if you met one person with autism, you've met one person with autism, and that was by Dr. Stephen Shore. This quote leads us to convey the differences in our communities today. You could have two people with the same mutation, but phenotypically present autism differently, which is essentially genetic diversity, the genetic difference between individuals. Now we're looking at CHLA gene, specifically the mutation that is presented in one allele in the heterozygous chromosome, but not in the wild type. With this mutation, it causes the highest genetic risk factor for autism. We are looking at CHLA gene mutation within the metaprefrontal cortex, which is responsible for the social and emotional behavior.
Now you may think, well, why is this important? It is important because neuroscience preclinical studies often lack genetic diversity usage, which is why we examine two genetically different strains. So from then, we looked at these strains that showed two different social behavior differences as well, therefore acting as a good model to emit a more realistic translation to the genetically diverse and communities that we have today. This model then allowed us to examine how genetic diversity and a CHLA mutation may affect the expression of CHLA in the heterozygous mice of both strains. So strain 1, or B6cc61, exhibits strong social behaviors, while strain 2, B6cc17, shows a deficiency in social behaviors. So then this allowed us to format our question: will genetic diversity impact the reduction of CHLA mRNA expression in heterozygous mice? From this question, we shaped our hypothesis. We hypothesized that there will be a range in reduction of CHLA expression in the heterozygous mice between strains due to genetic diversity. To test this hypothesis, we took our first approach, RNA-scope, which is essentially CDH8 fluorescence staining. So on the left, we have the wild type of our both strains and the heterozygous of both strains. And after RNA-scope, we took our slides and put them under a confocal microscope, which shows us our green CHLA fluorescence,
as well as our DAPI, which is our blue fluorescence, making sure that the CHLA reside within the cell nuclei, representing one RNA molecule. After our fluorescent imaging was completed, it was time to quantify our CHLA expression through Scamper pipeline, which leads us to our lovely graphs here. These graphs are representing the first strain, B6cc61. The graph on the left shows the percentage of cells that are expressing CHLA out of the total cells captured, which is expressing no significant change between the wild type and the head. Sorry. However, on the graph on the right side represents the mean of CHLA expression per cell, which shows actually a 40% reduction between the heterozygous compared to the wild-type mice.
So now for strain 2, B6CC17, the graph on the left again representing the amount of cells expressing CHD8 out of the total amount of cells. You can see that it starts to trend towards an increase of expression, as the same goes for the graph on the right, then representing the mean CHD8 expression per individual cell. This shows a 30% increase in expression, which might seem paradoxical because this is a behaviorally resilient strain. Although there are many regulatory or protective pathways that may influence and cause this increase. In all, this strain shows more overall CHD8 expression per cell and in the total cells in an attempt to protect itself from the mutation's reduction ability. So after seeing our data, we can say that our hypothesis was supported because there is a range in amount of CHD8 expression between the wild type and the heterozygous between the strains. So again, the socially dominant strain had reduced CHD8 expression by 40%, while the socially resilient strain had an increased amount of CHD8 expression by 30%. So there was a clear variability between strains caused by the CHD8 mutation. A contributing factor to this may be the genetic background differences. In all, the different molecular changes of the two strains due to the CHD8 mutation may be related to the differences in their social behaviors.
Which leads us on to our future work. Hopefully, we plan on using more mice strains that exhibit different social behaviors, increase sample size to increase experimental power, examine different ages to see potential development effects, use female mice, and rescue B6CC61 from CHD8 deficiency with hopefully using gene therapy.
And a big thank you to our lab on the 3rd floor, LA HIP administration, and our fellow LA HIP cohort and everyone joining us today. Thank you.
Thank you. Now I would love to welcome up Bianca and Allison. Allison attends Harbor Teacher Prep and Bianca goes to John Burroughs High School. Both shared a love for being in the lab with like-minded individuals and look forward to exploring the many fascinating niches of medical research. And they also both mentioned they loved having each other as lab partners, which I love. So it is my pleasure to welcome you both up to the stage. Hi, my name is Bianca Sherrick.
I'm Allison Paredes.
And we did our project on the role of neuregulin 1 and neuregulin 2 in wound healing of colonic epithelial cells. So our lab focuses on IBD, which is inflammatory bowel disease, and is characterized by chronic inflammation in the GI tract that is triggered by wounds in the epithelial barrier. So over here, you can see the epithelial barrier, which usually maintains a homeostatic separation between your microbiome and your lumen and your immune cells and your lamina propria. However, wounds in the epithelial barrier can allow bacteria from your lumen to leak into your lamina propria, triggering an immune reaction and inflammation. In most people, these wounds will heal and inflammation will die down. However, in genetically predisposed individuals, their immune system will overreact and it will create the cycle of damage and inflammation that we see in IBD patients.
Unfortunately, there is no cure for IBD, and viable treatments often disrupt patients' lives. To help improve IBD treatment, we are researching ways to promote wound healing in the epithelial barrier to stop invading bacteria from continuously entering the lumen propria and triggering the chronic inflammation. This leads us to the question of how can we stimulate wound healing in the gut lining to treat IBD?
So we decided to look at the potential of neuregulin 1 and neuregulin 2 in wound healing because they're both ligands of the EGF family, which is known to be regenerative in multiple organs. Also, we know that ERB3, which is the main receptor for neuregulin 1 and neuregulin 2, is essential for intestinal epithelium repair. This is proven by the graph to the right, which shows that mice without ERB3 receptors on their intestinal epithelial cells had a harder time recovering from colitis than mice with ERB3 receptors.
Furthermore, in the Fry lab, research indicates that M1 pro-inflammatory macrophages express neuregulin 1, while M2 anti-inflammatory macrophages express neuregulin 2. Notably, in the early stages of wound healing in other organs, macrophages first show an M1 phenotype and then switch to an M2 phenotype as wound healing progresses. Because of this, we want to know the significance of neuregulin 1 and neuregulin 2 expression of wound healing in the GI tract, as it is currently unknown.
This leads us to our research question. What is the role of neuregulin 1 and neuregulin 2 in the wound healing process of the colonic epithelial barrier?
We hypothesize that Neuglin-1 and Neuglin-2 will promote wound healing in colonic epithelial cells. The image on the right demonstrates this, with M1 macrophages secreting Neuglin-1 and M2 macrophages secreting Neuglin-2 to induce cell restitution and heal the damaged epithelial barrier.
To test our hypothesis, we cultured YAMCs, which are young adult mouse epithelial cells. Then we plated them and grew them until they were completely confluent, and then we starved them so they stopped growing.
Then we made wounds. 2 plates were stimulated with neuregulins, 1 with EGF, our positive control, and 1 control plate. We then took pictures of them at 0 and 8-hour time points to calculate for wound % closure.
We also did a qPCR on unwounded plates and stimulated with neuregulin-1 and neuregulin-2. After 2 hours, we checked for restitution markers.
This brings us to our results. For our wound healing assay, the images on the left show that both neuregulin-1 and 2 treatment reduced the size of the original wound made after 8 hours.
This is confirmed by our graph on the right, which shows that neuregulin-1 and neuregulin-2 stimulation accelerated wound healing similar to our positive control EGF. Also, our qPCR results showed that neuregulin-1 and neuregulin-2 stimulation upregulated claudin-4 and occludin expression, which are markers for the tight junctions that maintain the seal of the epithelial barrier. Also, neuregulin-1 stimulation upregulated MUC2 expression, which is a marker for goblet cells.
Triple factor 2, a cell restitution marker, was increased by stimulation of both neuregulin-1 and 2, but neuregulin-2 posed a stronger effect. And LGR5, an intestinal stem cell marker, was increased by neuregulin-2 stimulation.
This variation in qPCR results also supports our prediction that neuregulin-1 and neuregulin-2 play separate roles in cell regeneration.
To summarize our results, our wound healing assay showed that neuregulin-1 and neuregulin-2 stimulation accelerated wound closure, and our qPCR results revealed increased restitution markers in cells treated with neuregulins 1 and 2. Overall, this supports our hypothesis that neuregulin-1 and neuregulin-2 accelerate the restitution of colonic epithelial cells.
In the future, we would like to experiment with collonoids to see the effect of neuregulins in a more natural environment. We would also like to assess gene expression variation over time in our qPCR to see if there are any changes. And we would like to co-culture macrophages and epithelial cells to see how they interact.
We also want to say thank you to the Fry Lab, our mentor, Dr. Vinny John, and LAHIP for this opportunity.
Thank you. Thank you. Now I would love to welcome up Lola and Melissa. Lola attends Camps High School and Melissa attends Warren High School. Both stated that they love to work in the lab to receive hands-on experience, and they love the connections that they were able to make with LAHIP alumni, PhD students, fellows, and PIs from all different research disciplines. I would love to welcome Lola and Melissa up to the stage.
Hello, everyone. My name is Lola Obashde.
My name is Melissa Cantrola.
And today we will be presenting our research on understanding osteopontin's role in the efficacy of anti-CD19 CAR T cells against B-cell leukemia. So B-cell acute lymphoblastic leukemia is a very aggressive and common form of cancer located in the blood and bone marrow. The image on the top right shows a typical production of blood cells in the bone marrow. In patients with leukemia, there's an abnormal production of these cells. Leukemia cells, also known as blasts, multiply uncontrollably and crowd out normal cells in the bone marrow. And you can see this displayed in the bottom images comparing normal cells to leukemia cells. Despite much progress in the field, survival rates have plateaued, and current therapies such as chemotherapy have long-lasting and toxic side effects. Because of this, new therapies for leukemia are needed. One of the most promising and novel therapies is CAR T-cell therapy. In this therapy, T cells from the patient's immune system are genetically modified with CAR receptors to easily recognize and attack cancer cells that have a specific marker on their surface called CD19. In the image, you can see the CAR T cell binding to the cancer cell through the CD19 marker. CAR T cells will attack the leukemia cells, which results in the death of the leukemia cell. The major conflict with this therapy, however, is that 50% of patients relapse after 1 year. This is because of problems within the tumor microenvironment, allowing leukemia cells to evade the CAR T-cell therapy treatment.
So So when trying to find a contributing cause to this problem, we aim our attention to the tumor microenvironment where leukemia cells reside. In here, there is a naturally occurring protein called osteopontin, and if you look to the image on the right, you can see when osteopontin interacts with the leukemia cells, it increases the NF-kappa B gene expression in the cell and upregulates the PD-L1 surface expression. When this cell interacts with the CAR T cell, which has the PD-1 surface expression, it inactivates its function and turns off the CAR T cell activity. So with this said, we're wondering, is there a similar mechanism that is working in the CAR T cell therapy? So from here, we're saying that osteopontin will inactivate CAR T cells, weakening the efficacy of CAR T cells against leukemia. And we're going to figure this out through testing if osteopontin induces the NF-kappa B gene pathway and upregulates PD-L1 surface expression in leukemia cells. Our first approach towards this is by doing Western blotting. We cultured the leukemia cells in stroma with osteopontin, then we suspended the proteins to get our proteins of interest, putting them into a gel and then electrotransferring that gel. From there, we blotted that gel onto a paper and probed that membrane with antibodies. Then we imaged it in a machine. The purpose behind this was to check the levels of activated NF-kappa B on BALL after osteopontin treatment. As you can see in our results here, our treated cells, which are the 5 and the 50, experience a much more intense blotting in comparison to our baseline control and our positive control interferon gamma, which is a known inducer of NF-kappa B. You can observe this trend also in our P65 and P50, which are components of the phosphorylated P65 pathway. And if you can look to the right, you can see that in our graphs, there is a quantified version of the relative intensity that observes a similar pattern where the treated osteopontin cells have an increased relative intensity, both in the phosphorylated and unphosphorylated.
Next, we wanted to measure the activation of PD-L1 expression after treating the cells with osteopontin. We separated our cells, stained their surface markers, and conducted flow cytometry. And flow cytometry is a laser-based technique used to detect and analyze cell characteristics. So after conducting flow cytometry, we created a histogram of the raw data, and the x-axis of the histogram represents the level of intensity of PD-L1 surface expression. And you can see that the orange and red curve representing samples with osteopontin moves more towards the right of the x-axis compared to the blue curve, which represents the control. And this result helps us determine that osteopontin substantially increases PD-L1 surface expression. And you can also see this data displayed in the bar graph seen on the right, showing the amounts of PD-L1 expression in each group. One bar represents our control, one IF gamma our positive control, and one osteopontin. In the bar graph, you can see stars at the top of the osteopontin and IF gamma bars. And this star signifies that these results are statistically significant as their p-value is less than 0.05. This further confirms our hypothesis that osteopontin does in fact trigger and upregulate PD-L1 surface expression.
So to conclude, we were able to say that osteopontin induces levels of activated NF-kappa B in the leukemia cells, and it upregulated the PD-L1 surface expression on these cells. Our next step from here is to show that osteopontin inhibits CAR T cell function because osteopontin may play a role in inhibiting CD19 CAR T cells against BALL.
Thank you to our mentors, CHLA faculty, and LA HIP, and thank you for your time.
Now, I would like to welcome up Paul and Robbie. Paul attends Mark Kreppel High School, and Robbie attends Azusa High School. Robbie and Paul both really appreciated the work they were able to do in the DIAS lab, and I look forward to implementing those skills in the future. Welcome you.
Hello, my name is Robert Vasquez.
Hi, my name is Paul Galarza, and we worked under the mentorship of Samantha Stack and Dr. Dias.
And today we're going to be talking about neuronal markers in the mice brain, specifically tracking IBA1 and Claudin-5 through development.
In a city, there are bustling streets, tall towers, and big intersections all working simultaneously. This type of complexity is also found in the brain, with its numerous pathways and regions. Just like the city, the brain requires certain mechanisms in particular to ensure that things flow efficiently and effectively.
Oh. And as the city needs a trash collector, the brain needs microglia to help regulate any unwanted material.
Like the city buildings have their security guards, the brain has its blood-brain barrier, which regulates the substances that can go through the blood vessels and into the brain.
We just talked about a fully matured brain, but a main focus in the DIES lab is how the developing brain is affected by early life adversity. The first step in this process is to look at the status of microglia and the blood-brain barrier through development. One of the main functions of microglia is synaptic pruning, which is the process of removing any unnecessary neural pathways. Microglia is also a key contributor in neural optimization. These are some of the functions we are primarily focusing on. To stain for microglia, we use a primary called IBA1, which is a marker of microglia. This is helpful to look at microglia under the microscope.
Now for the blood-brain barrier. As I mentioned, it works as a filter between the blood vessels and the brain cells. Looking here at this diagram depicting a blood vessel that was sliced down, lining it are these cells called endothelial cells, and in between these cells are tight junctions, where tight junction proteins reside and attach to the cells, tightening up the and restricting the passage. We studied the tight junction protein claudin-5 and used that as a marker for the blood-brain barrier. To detect microglia and the blood-brain barrier, we used immunohistochemistry. We took the mice brains that were fixed in PFA to the cryostat and sectioned them at 35 micrometers and mounted them onto slides. Then for immunohistochemistry, we applied a primary antibody targeting either IBA1 or claudin-5, which attached to the tissue. The next day, we applied a secondary antibody, which attached to the primary, but this time it had a green fluorophore, which allowed us to see the staining under the microscope. At the microscope, we focused on two brain areas associated with learning and memory, the prefrontal cortex and the thalamus. And this was because the DIES lab is interested in understanding how development impacts these regions.
Before we get to our questions, we remind you all again about how the lab is mostly interested in the developing brain. So the big question, does the expression of microglia in the brain change across development? So after reading some literature, I hypothesized that due to the need for synaptic pruning and optimizing neural circuits that change over time, the expression of microglia will change across development.
For the blood-brain barrier, my research question was, is the blood-brain barrier comparably developed in brain regions associated with learning and memory across development? And since research shows that the blood-brain barrier is functionally mature at birth, I hypothesized that the developing brain will express markers associated with the blood-brain barrier in learning and memory regions to a level that is comparable to the adult brain.
So for our images, the prefrontal cortex is shown on the top, while the images of the thalamus are shown on the bottom. Based on the data, we observed a greater microglial density in P23 prefrontal cortex compared to the P21 prefrontal cortex, as indicated in the green fluorescence. As a side note, postnatal days are represented by P. Furthermore, this data shows significance in the prefrontal cortex, but not in the thalamus.
And here are the images that we took for claudin-5, which is the green fluorescence. We looked specifically in the prefrontal cortex. The images were taken at 3 different ages, with P21 and P23 representing more adolescent mice brains, and P60 being the adult mice. Seeing here, through these 3 stages, the expression of claudin-5 around what we can assume are the blood vessels is very comparable and stays consistent.
From the data previously mentioned, this further supports my hypothesis due to the need for synaptic pruning and other neural optimizations. For our further direction, we would increase our sample size to obtain more accurate results with the help of the DIES lab.
For the blood-brain barrier, the data support my hypothesis by showing comparable expression of blood-brain barrier marker in the prefrontal cortex through development. For future directions, now that we have used the antibody for claudin-5, our lab can now focus on learning how to quantify claudin-5, specifically since it is measured by intensity and not count. Also, the DIES lab looks at intergenerational effects of experiences, including certain diets. So the next direction would be to look at the changes in microglia and blood-brain barrier expression when mice are exposed to a high-fat, high-sugar diet while in utero.
We would like to give thanks to all of our lab members, mentors, and Elliott family for their tremendous support.
Now, I would like to welcome up Isabella and Serena to the podium. Isabella attends Laces High School and Serena attends Harbor Teacher Prep. Isabella says that her favorite thing about LA Hip was she loved meeting new people with a shared interest in science and looks forward to pursuing a career in medicine. Serena also hopes to see where she ends up in the medical field and also enjoyed the unique experiences here. I would love to welcome them to the stage.
Hi everyone, my name is Serena Hennepin.
And my name is Isabella Martinez.
And our project is about smart flares, illuminating pathways in Wilms tumors research.
The Wilms tumor is a rare pediatric cancer that's caused by a presence of cells overexpressing 6-2 after birth.
So, as Isabella mentioned, 6-2. So, what is it? It is a transcription factor that aids in the development of the nephrogenic niche in the embryonic kidney. So, basically, it helps with the kidney's development. To the right, that bean-shaped thing is the kidney, and next to that's the nephrogenic niche. As you can see, all those red dots are cells expressing 6-2, which is what helps it develop. And normally, they are exhausted before birth because its mission's done and the kidneys are developed, so they're no longer needed. But in the rare cases that 6-2 remains after birth, the cells continue to proliferate in a crazy dispersed manner, as you can see in these bottom Wilms tumor images, which is why the Wilms tumor is developed. And in all, these cells expressing 6-2 are difficult to isolate, which is a roadblock when trying to treat this pediatric cancer.
So with all of that in mind, we hypothesized that we're going to isolate cancer stem cells that express 6-2 with smart flares, and the final goal being to study the mechanisms that regulate Wilms tumor development.
So Isabella mentioned smart flares, and what are they? So they're a technological tool that we created over the course of 3 days. They're composed of a gold nanoparticle, capture strands, and reporter strands.
And also, by the way, this was very big of a surprise to us, but the gold nanoparticle does not look like a gold nanoparticle. It's like this red liquid. We were shocked.
And so the reason why we're developing them is to isolate the cells expressing 6-2 through live RNA detection. And once probed, the smart flare infiltrates the cell cytoplasm, and with the strands, they bind to the targeted mRNA, which is the 6-2, and release a fluorescent signal to the probed cells to allow us to visualize the isolation. And this entire process ensures that the cells are not damaged and even allows us to reuse the smart flares after this experiment.
All right, and you see here on the left is just kind of a photo of the Wilms tumor cells, just all of them, not just the ones expressing 6-2. So then we use a smart flare, and you see on the right here, this is with the smart flare, so the cells expressing 6-2 are now lighting up, and we can actually see where they are and therefore isolate them.
So after the smart flares were probed and they identified the cells expressing 6-2, as you saw in those images, we gathered the total cells that we cultured and ran them through a FACS sort data machine. And with this machine, it took the total cells provided and separated them into two categories, into the 6-2 positive cells and from the negatives. And as a result, we found that 63.6% of the cells that were expressing 6-2 positive, which was very successful.
And after we had that FACS sorting, we used a Western blot. And so basically the Western blot tells us if there's a protein expression of 6-2 in the cells that we actually isolated. And you can see here it's super, super dark, meaning there's a lot of 6-2 presence. So while we're doing the project, there were a couple difficulties and roadblocks, but honestly, that's just kind of what research is. And I think everyone has learned that research is kind of just there's issues with it. And it's also a testament to your patience and your dedication to research and science. So some issues that we had personally were we were trying to grow the Wilms tumor cells, and we were using old media. And once we realized that, the cells no longer were dying, but it definitely was a roadblock. And then also the concentration of the DNA probes for the smart flare, we kept on getting sent the wrong amount. But once we were able to solve that, we found out that smart flares are a useful tool for Wilms tumor and also probably other therapies in the future.
And we'd like to acknowledge our entire lab for helping us with this project, LA Hip, my partner, and thank you.
All right, now I would like to welcome up Charlie, Jenny, Bernice, and Seth. Bernice attends West High School, and then Seth attends Miracosta, Jenny attends Orthopedic Hospital Medical Magnet High School, and then Charlie attends Grover Cleveland High School. All students were very happy with the work that they did in the lab, and they look forward to seeing how they're able to implement that in the future. It is my pleasure to welcome up all of you.
Hello, everyone. My name is Bernice Kemji.
My name is Charlie Perez.
My name is Jennifer Ramirez Flores.
My name is Seth Pickens.
And we did our research on the epithelial response of inflamed intestinal tissue in a mouse model of IBD.
So, IBD, or inflammatory bowel disease, is characterized by inflammation of gastrointestinal tissue and affects around 5 million people worldwide. With current treatments, only around 40% of patients report symptom relief, but it's important to note that within that population are individuals who may have experienced symptom relief but have since then fallen out of inflammatory remission. Some common treatments used today include biologics, corticosteroids, aminosalicylates, along with others. These treatments, while somewhat effective, are often taxing on the body and have many adverse effects. Another thing to note is that current treatment options target the immune response in IBD patients, when the common issue across all forms of IBD is that the inflammation often damages the epithelial layer of the intestine. Therefore, we believe that understanding IBD on a cellular level would help us develop treatments that would rather work to restore the epithelium rather than alter the immune response. We also believe that studying the cellular response of the intestinal epithelium at different stages of colitis may help us better understand the disease. When observing the cellular composition, we're looking at 4 different cell types: goblet cells, tuft cells, enteroendocrine cells, and stem cells. Goblet cells secrete mucin, which is a compound of mucus, and they aid in lubrication and protection. Tuft cells are chemosensory cells that release biologically active mediators. Enteroendocrine cells release hormones in response to stimuli, and stem cells are proliferative cells that are the source of all cells in the epithelium. And it's also important to note that if you'll pay attention to this picture here, these are how the cells are situated in the colon. However, in the small intestine, these sections here, known as crypts, are much smaller, and these sections here are much longer, and they're called villi.
So with this background information in mind, the research question we came up with was, does chronic inflammation change the cellular composition of the intestinal epithelium and impact pro-repair responses?
Some patients with IBD were observed to have defects. Therefore, our lab decided to model this using mice. And in our lab, we had 4 groups, as you can see here. We had a group with no colitis, a group with IL-10 induced, a group of mice in 2 weeks and 4 weeks in recovery. And essentially, the small intestine and colon were dissected from the mice and placed into slides, which we stained using a primary and secondary antibody. And we were able to capture these Swiss rolls. And the outermost region here is called proximal, the middle is medial, and the innermost is distal. And we were approximating about 30 crypts per region. And former LA HIP students, Nina and Itzel, worked on qPCR. And essentially their qPCR analysis data suggests that the cell populations are changing as they measure the RNA with qPCR. And we wanted to test the results more directly through immunofluorescence, which targets the protein. So here is an example of the colon immunofluorescence staining. The green is for epithelial cells, the blue for goblet cells, and the red for proliferating cells. We had targets which were the nucleus, the epithelial cells, proliferating cells, goblet cells, and tuft cells. And only our goblet and tuft cell stainings were partially successful. We don't have enough images for significant quantitative data. And then this is for a small intestine. The green is for epithelial cells, the blue for tuft cells, and the red for proliferating cells. And our targets were the same as nuclei, epithelial cells, proliferating cells, goblet cells, and tuft cells. And we only had time to stain for DCLK positive cells, tuft cells, and we focused on analyzing
Ki67 positive proliferating cells, and DCLK positive tough cells.
This is an example of our proliferation staining. So all these bright red cells are the proliferating ones that are Ki67 positive, meaning that they have the marker that we're looking for. As you can see in the no colitis, our control, there seems to be the lowest amount of proliferation, and colitis seems to raise that, and we can see that raising, persisting throughout the whole process. Our quantitative data backs up our visual observations. After we counted each of the Ki67 positive cells, we found that it was lowest in our control, and after colitis, it rose, which seems to peak after 2 weeks. Once we move on to the small intestine, we see a similar pattern, but instead of a peak at 2 weeks, it seems that the growth keeps continuing after colitis has happened. And in our DCLK marker, which measures our tough cells, which react to infection and whatnot, we can see that it's normal for the first 3, and it peaks. It doesn't peak. It rises at 4 weeks after recovery. So we're still trying to figure out how that happens.
So from our results, we found that there was generally increased cellular proliferation in both the colon and the small intestine, which indicates a possible change in the cellular activity and composition of the epithelium in response to colitis. We also found that in the colon, cell proliferation peaked in mice that had been recovering for 2 weeks from colitis, and we believe that's due to the epithelium trying to get back to homeostasis. With no colitis, there's a normal amount of cellular proliferation, and when colitis happens, the proliferation ramps up to get the epithelium back to homeostasis. When homeostasis is reached, the epithelium returns to normal cellular proliferation levels. Also, we see in mice that had been recovering for 2 weeks, the proximal region of the colon seemed to have the most cell proliferation, but we're not sure why. In the small intestine, the number of tough cells, or DCLK1 positive cells, increased in mice that had been recovering for 4 weeks, indicating that they may be important in the epithelial recovery process. Although the cellular proliferation we've seen indicates a possible change in the cellular composition of the epithelium, further staining has to be done in the future to identify what cell types are dividing and contributing to recovery. We hope that with more research, more treatments for IBD will focus on the epithelium instead of dialing down on the immune response. And lastly, to our amazing mentors, Dr. Fernandez and Dr. Schumacher, and to everyone who made LA HIP possible, and for you guys for listening, thank you.
Oh, I see. Sorry. There. That's good. Okay. Thank you. Now I would like to welcome up our final group, Caitlin and Anna. Caitlin goes to Monrovia High School, and Anna goes to Bravo Medical Magnet High School. Both have stated that, well, Caitlin specifically says that her experience in LA HIP, she got to apply a lot of fundamental information to what learning in a lab was like. In her future, she hopes to attend university and later become a physician. Anna also enjoyed the professional setting of LA HIP and hopes to become a pediatrician one day. Okay, I would love to welcome our final folks up.
Hello, everyone. My name is Caitlin Morrison.
And I'm Ana Villatoro, and we have been working on optimizing podocyte differentiation of amniotic fluid progenitor cells.
So for those of you who don't know what amniotic fluid progenitor cells are, let's get to know one. So progenitor cells are derived from amniotic fluid stem cells. These stem cells are then differentiated into progenitor cells, which then can differentiate into many organ-specific cells. And like I said, they are from the amniotic fluid, which is the nutritious fluid that surrounds the fetus in the womb during pregnancy. And in our lab, we have been specifically working with kidney progenitor cells that are interested in becoming podocytes.
So what is a podocyte? As we know, kidneys are blood filtering organs that keep proteins that we need inside of our body, excreting the waste. And specific cells that do this are known as podocytes. Podocytes are terminally differentiated, meaning we are only born with a certain amount of them and they cannot proliferate. A specific protein that is important for a podocyte is known as WT1, and its expression is controlled by HIF1 alpha.
So the issue that we have been studying in our lab is called Alport syndrome. Alport syndrome is a genetic disease in which a patient lacks the production of collagen type 4. Collagen is the binding agent of podocytes, so without it, podocytes are damaged and then lost in the urine of Alport syndrome patients, which can be seen In this diagram, as the podocyte is detaching from the structure below. A loss of podocytes results in a loss of valuable proteins and then eventually kidney failure.
So we have been working on the proliferation of these cells. We have been using two cell lines, healthy HAKPC cells and Alport syndrome cells. And we have been expanding and differentiating them, adding cobalt chloride to see if that will better their differentiation process.
To check our results, we use immunohistochemistry, in which we fix the cells and then stain them with DAPI and F-actin. The purpose is to see the cell structure as well as the location of possible key marker proteins within the cell, or if there are any presence at all. So why cobalt chloride? Cobalt chloride mimics hypoxia, which is the removal of oxygen from the culture dishes and creates a cellular response. Our predicted cellular response was an increase in WT1, and we think this because in previous experiments with Wilms tumor cells, the use of cobalt chloride did increase the production of WT1. So we hope the same is true for podocytes.
So with that information, our research question is, will inducing hypoxia with cobalt chloride have the same effect on podocyte differentiation as Wilms tumor cells? And our hypothesis is that we can increase WT1 in podocyte development with cobalt chloride to better podocyte differentiation.
So now that brings us to our results. And like I said before, we stained with DAPI, which stains the nuclei of the cell, which are these blue figures right here. The pink on top is the specific protein or antibody that we stained for. So as we can see under WT1, there was an increase in production in the treatment column compared to the control. And the same is true for HIF1 alpha. The presence was much more emphasized in the treatment group compared to the control. And this is important because, again, WT1 and HIF1 alpha are podocyte key markers and are important for the podocyte identification. And then we stained with F-actin, which stains the cytoskeleton of the cell so we can see the cell structure. As we can see in both the control and the treatment group, there are podocyte-like characteristics, such as these foot processes, which are very important for attachment in vivo, as well as even in the treatment group, we can see a double nuclei here, which is very common in podocyte differentiation processes.
So for our Alport syndrome cell results, they are a little bit different because Alport syndrome cells are damaged. As we can see in our control, we see there is more WT1, although the problem is it is outside of the nuclei, which is not where it's supposed to be. That is where the cobalt chloride comes in and kind of controls that and keeps it inside of the nuclei. Same thing with HIF1 alpha, we can see that there is a bit more of the expression there, but it is outside of the nuclei, and then the cobalt chloride kind of comes in and betters that. And for the F-actin stain, we couldn't tell if that really improved the structure, but we can tell that it does have podocyte-like characteristics.
So in conclusion, our hypothesis was correct. The cobalt chloride brought out the presence of podocyte key markers significantly compared to the control. And in spite of our success, we did hit many obstacles along the differentiation process with dead cells and variation in method, and as well even in the results period with Western blotting attempts.
So for future work, I hope that our work can help with therapies for kidney disease patients, and we would like to further confirm our results by inducing hypoxia in other cell lines and other cell differentiation processes. We thank our mentor, our PI, and everyone in the GoFar lab that helped.
We also want to thank Danny, Emmanuel, and Mateo for all their hard work, as well as LA HIP for the opportunity. Thank you all for listening.
All right, well, that concludes our scientific presentation. I just, again, want to congratulate the students on all the amazing work they did. About 6 months ago, my sister was cleaning out some boxes that were left over in our parents' house from when we were kids, and found some photos from an old science fair that I was at in high school. I looked at the project I did, which was some dumb, out of a kit electronics thing that you follow the numbers by. And I was thinking about that all summer as these guys were doing this work. I couldn't have done any of this stuff then. So they're really amazing. And I did forget to thank one really important group of people at the beginning, which is the families and friends who have supported these students in wanting to do this program. I know it's not trivial to get them here on time, have them be gone all day, all summer. They owe you a round of applause for helping them make this happen for them. So. All right. That said, I think it's time to go out and go and have a reception outside. Danny, anything else we got? All right. Once again, the class of '24.